Welcome back to the crypts. If you've watched our lesson on the common pathway of the complement cascade, you'll see some familiar friends over there giving each other a high five, which also that's a weird thing to put on your tomb. Anyway, it means one thing. We're still talking about complement, but don't worry, I promise this will end soon. In this sketch, we're going to explore the 3 different ways complement can get activated. In our lesson on the complement cascade, we learned that all roads lead to the common pathway. But if you don't know where you came from, you can't know where you're going, or something like that. So let's talk about each of these activation pathways. As a quick refresher, each complement activation pathway converges on the big moment when C3 gets cleaved into C3A and C3B. And after that, it does a whole bunch of stuff that we talk about in our other lesson. But safe to say it's magnificent, Anyway, the first pathway we'll talk about is the classical or antibody-dependent pathway, and we'll symbolize that with this row of classical columns sporting an antibody-shaped nest. Of all the 3 pathways, this is the most complicated one, so don't worry, it'll all be easy after this. The classical pathway is all about C1. We'll symbolize that with our crow with one giant eye. Get it? One seeing C1. C1 is a giant complex made out of many proteins, the names of which you could learn, but also life is short, and in any case, we're going to stick to the basics here. C1 on its own has an inhibitor molecule attached to it, which means it can't take off and fly free, just like this crow can't due to the bands on its wings. However, if those inhibitors do come off, C1 can do some glorious things. So how do they drop those pesky blockers? Well, that's where the antibodies come in. Remember, this is the antibody-dependent pathway. IgM is the biggest of all the antibodies, and it's made up of many individual immunoglobulin subunits. Having all these subunits together is super useful for bringing multiple C1 complexes together. And for completeness sake, let's just note that some IgG molecules can do this as well. So we'll have another one-seeing crow land on this IgM-shaped nest to really highlight that IgM is great at bringing together multiple C1 complexes. And what do you know? When 2 or more crows come together, that's when the fun starts. As you may have guessed, bringing together multiple C1s is the trick to ditching those inhibitor molecules.
Fly, friends, be free.
Gonna hope our production team changed up that weird voice. Anyway, once C1 is, uninhibited, it can start a cascade that eventually leads to the first step in the common complement pathway. And again, just to beat you over the head with this, that's when C3 is cleaved into C3A and C3B. To illustrate this, we'll have our now airborne crow knocking over these classical columns that knock each other over like dominoes to land inside the common room. That's a cascade if I've ever seen one. And look at that. That's one activation pathway done. And lucky for everyone, the other 2 are super easy to remember. The next pathway we'll talk about is the alternative or antibody-independent pathway, symbolized by this turning, spiraling path. This is maybe the easiest pathway to remember because C3 just spontaneously cleaves itself into C3A and C3B. Just like this 3-seeing crow is spontaneously bursting into our 3-seeing crow with B glasses and 3A jewels. And that's it. That's the whole alternative pathway. Not much to it. Finally, we have the lectin activation pathway. Mannose-binding lectin, or MBL for short, is a protein present in all human bodies. It recognizes and binds the mannose carbohydrate molecule that's found on the surface of many pathogens, but crucially not found on the surface of healthy human cells. And to make sure you remember the function of MBL forever, this statue is lecking a man's nose. Oh yeah, we're geniuses. Once MBL binds mannose on a pathogen, it activates a protein called MASP, which we'll symbolize with this MASP, an asp with an M-shaped tongue. In case you don't do the New York Times crossword every day, an asp is a type of snake. And see how the asp's tongue has slithered over to touch our 3-seeing crow? That's because MASP, our MASP, goes on to clip C3 into C3A and C3B, which means we're right back to the common pathway. That actually wasn't so bad, right? In fact, this lesson is so short and sweet, we don't even think we need to recap it. All you need to remember are our dominoing classical columns, our turning, spontaneously bursting crow, and lecking a man's nose with an M-asp. Just usual stuff. And that's it for complement. I've had a great time down here in the complement crypts with you. But it's a little bit spooky, so let's get out of here and back into the daylight.